Hello friends, today we are going to discuss about active transport. Okay, in this we will first discuss about primary active transport. That is 1 degree active transport or primary active transport. When a substance or an ion is transported across cell membrane, this is your cell membrane, and when any substance or any ion is transported across the cell membrane against a concentration gradient or electrical gradient with the transport system directly breaking down ATP molecule to provide the energy, it is known as primary active transport. Okay, primary active transport involves the use of your ATP. We will discuss detailed mechanism behind this primary active transport. Now, the most common primary active transport is your sodium potassium ATPase. This is the most common active transport. Now, this sodium potassium ATPase is responsible for maintaining, this is your cell, okay, and this is ECF, extracellular space. So this sodium potassium ATPase is responsible for maintaining high intracellular potassium level and low intracellular sodium concentration. Okay, so high potassium concentration and low intracellular sodium concentration. This is the basic function of the sodium potassium ATPase. Now, the energy which is required for the movement of this sodium and potassium is given by ATPase, ATP. Okay, hydrolysis of ATP will occur, and this will lead to the release of energy which will transport ions against the concentration gradient. Okay, now coming to the mechanism behind the transport. So basically, sodium potassium ATPase is a protein with alpha and beta subunit. It has two subunits, one is alpha and one is beta. This is your, suppose this is your cell membrane, okay, and this is your beta subunit, and this is your alpha subunit, okay, and this alpha subunit has binding site for, this is the intracellular portion, okay, this is your intracellular portion, and this is your extracellular portion. So intracellularly, it has 3 binding site for sodium and 1 binding site for ATP. Okay, so the alpha subunit has intracellular binding site for 3 sodium plus and an ATP molecule, whereas on the extracellular side, on the extracellular portion, it has binding site for 2K plus. Okay, so this is the basic structure of the sodium potassium ATPase. Now coming to the functioning of this. When, let's again draw the same diagram. This is your alpha subunit, okay, and this is your beta. When 3 sodium and 1 ATP molecule bind to the intracellular site, this is the 3 sodium and 1 ATP. So when ATP bind here and sodium bind here, when 3 sodium ions and 1 ATP molecule bind to the intracellular sites, the ATP breaks down. The ATP breaks down and it will break down into ADP plus inorganic phosphate, and this phosphate will phosphorylate an aspartic acid residue of the alpha subunit. Okay, this is your alpha subunit, okay, alpha subunit, beta subunit, and this phosphate will phosphorylate the aspartate residue of your alpha subunit. Okay, so this produces, the phosphorylation of aspartate will produce a conformational change in which 3 sodium is extruded out of the cell. Okay, 3 sodium is extruded out of the cell after the phosphorylation of alpha subunit, after the phosphorylation of aspartate residue of your alpha subunit. Okay, now then after this, after the extrusion of the 3 sodium, 2 potassium will bind to the 2 potassium, one here and one here will bind to the extracellular site. Okay, and this will lead to the dephosphorylation of the aspartate residue. Okay, binding of 2K plus will lead to dephosphorylation. Okay, and once dephosphorylation will occur, then 2 potassium plus will be transferred inside your cell. Okay, so this is the basic mechanism how sodium potassium ATPase will operate. Now coming to the clinical of the transport. So the drug, that is your digitalis, and one more drug, that is ibuprofen. These two drugs mainly bind to the receptor on the outer surface of sodium potassium ATPase. Okay, it will bind to the outer surface of the sodium potassium ATPase. I mentioning the outer surface of the sodium potassium ATPase, and it interferes with dephosphorylation step. Okay, because after binding of your potassium to the, suppose this was your alpha subunit, this was the potassium site, one potassium here, one potassium here. As the potassium binds to the extracellular site, dephosphorylation of the aspartate residue will occur, which will lead to the transfer of potassium inside the cell. Okay, so digitalis and ibuprofen, these two drugs will interfere binding of potassium to the outer surface. So dephosphorylation of your aspartate residue will not occur. Okay, so this is the basic mechanism behind the functioning of your digestive.